All right, so when we are,
Therefore,
so when we
it's a bad idea.
take notes in class, what we'll do is I have a presentation up there that looks like your packet. However, you have a bunch of blanks in your packet, and I will fill them in up here on the projector. So we're talking today and in this first unit about what we call a scientific method. And I know you've talked a little bit about it before, and you guys learned about hypothesis and so forth. We're going to get into some more detail. We're going to talk about variables and some of the steps and so forth. So that's sort of where we're going with this. So you have in the beginning of each note packet, usually, there's a list of vocabulary. We're not like going to go over it right now, but you have that kind of you refer to as we're going through the unit to look back. Those are some really important key terms for our unit. So that's what those are. So let's start off just by looking at this example here. So a student studied the effects of red and blue light on plant growth. To do this, she set up an experiment and grew 2 plants under the conditions that are shown in the drawing. And at the end of the experiment, plant 1 grew 10 centimeters, and plant 2 grew 5 centimeters. And so she concluded plants grow better under red light. So if we look at that and how the experiment was set up, what do you think about her conclusion? Is it correct? Is it incorrect? Is it appropriate? Tell me something about her conclusion. Sean?
It's not very accurate
How come?
because the variables are different.
Like what?
The water.
Okay. What was she testing? Or what did she say she was testing? Or think about? You see it? Right. She was trying to test the effects of blue and red light. But like Sean was saying, well, she gave him different amounts of water. So what would that do to her conclusion in this experiment? Abby?
It would make it so that she was not just testing the different colors of light, because if they aren't giving the same amount of water, if they aren't the same plant, if they aren't at the same temperature, then you're going to get a different result.
Okay, excellent. Yeah, so she can't be sure of her conclusion because the experiment was not set up correctly. Okay? So how would she improve upon it? So you're correct. This is not a reliable. The word we use is a valid experiment. This is not a valid experiment. So how could she improve upon it? What changes should she make in order for this to be a valid experiment? Zanette?
Give them the same temperature and give them the same amount of water.
Okay. Anything else? Addis?
Allow them the same amount of time.
Okay. Anything else, Jack?
Grow the same plant.
Okay. Yeah. So in setting up this experiment, what should be the only thing different in order for this to be a good experiment? Sean?
The color of the light.
Yes. So when we set up an experiment, to do it properly, we want to have one thing different between our groups, and we want to try to keep everything else the same. Because the way it's set up now, why did plant 1 grow double plant number 2?
Because it had more temperature.
Do we know that? We do? We know that's why? Zanette? Do we know that's why?
No. Well, yes, because it has more time, it has more water.
So which thing allowed it to grow double the size? I don't know if that's true.
The time.
You think so? Do you know this, or you're just sort of guessing?
I don't know this for sure, but I personally believe it's because it's a completely different type of plant.
Okay, but I don't want to know what you think. You guys are arguing what you think, what you believe. Can we know for sure? We don't know which one changed
Because it wasn't
the height.
valid experiment.
Right. So maybe it was the water, but we don't know for sure. It also could have been the type of plant, or it could have been the light, or it could have been the amount of time. But we don't really know because it's not a properly designed experiment. Okay, so you guys are right. We want to make sure everything is the same, except for the variable, we call it, that we're testing. In this case, it was the color of the light. If we had set it up this way, and we found the same results, then could we be more confident of the conclusion? Yes. All right, so this gets to some of the most important concepts of how we design a proper, valid experiment. So we are going to list 6 steps to the scientific method. And this is one version of the scientific method. It could vary. Different scientists might not use this exact same thing. But for our purposes, these are the 6 steps. We're going to have you guys remember what they are, what they mean in the correct order. So to help you remember them, we have a little saying. You could remember this saying, people really hate elephants on cars. Or if you remember this picture of Mr. Arcuri standing on the car, that will help you to remember the steps. Okay? People really hate elephants on cars. So each letter of the word stands for one of the steps. So the P stands for problem. We'll talk about what these actually mean in a minute. The R stands for research. The H stands for, if you can guess this one, correct, hypothesis. Anyone guess the E? Experiment. Colin, you know what the O is?
Observe, or like observation, yeah.
Yep. Passion? Yeah, and then the final step is the conclusion. So problem, research, hypothesis, experiment, observation, conclusion. You're going to have to memorize those in that order. Just like I said, it shouldn't be too difficult using this saying. So let's talk about what they actually mean. So when we're talking about the problem or the observations, what does that really mean when we're doing a science, when we're trying to learn something using the scientific method? So we'll start with the problem. Scientists are constantly asking questions. And in the problem step, that's when you pose a question, trying to figure something out. In the experiment we just were talking about, the problem would be, do plants grow taller under red or blue light? That was the question the scientist was asking. Before you can set up your experiment and so forth, though, you need some background knowledge. For example, in those plants, you need to know what kind of conditions they need to grow, what type of soil they should have, how much water. Those are things you would learn by doing some research before, gathering information that's going to help you to prepare your experiment. Once you've done a little bit of research, you have some background, it's time to develop or formulate your hypothesis. Which again is not any longer, we're not going to say educated guess anymore. We're going to call it a prediction. You're going to predict what you think will happen in your experiment. So in that experiment, the hypothesis might be that plants will grow taller under red light. Or maybe it would be that they grow taller under blue light. Or maybe that there would be no difference. Your hypothesis is yours, and it's set up before you actually do your experiment. So you don't know whether it's going to be correct or not until the experiment's happened, until the experiment's finished. So once we have our hypothesis, it's time to set up our experiment. An experiment is just a procedure we follow that's going to test our hypothesis. That's the point of an experiment. So the experiment we were just talking about. You would set up the plants, like one in red light, one in blue light. We give them the same amount of water, same plant, same amount of time, etc. And then we would move on to the next step, which is to observe our experiment. When we say observe, we might mean observe it with our eyes, but we also mean measure certain things, use a tool to collect data in our experiment. In the experiment we were just talking about, our observations would be measuring the height of it. And then finally, once you've collected your data, it's time to arrive at a conclusion. And your conclusion, you look at your data, look at your hypothesis, and you make a decision. Was your hypothesis supported by the experiment, by the observations, or was it not supported? So that's what the steps mean. Let's look at another example. Let's imagine an experiment when we're going to ask the question about rats and vitamins or mice. It's actually a rat. Let's say we want to answer this question. Do mice gain weight if they are given extra vitamins? That's going to be the problem we're going to think about. Do you think you could set up an experiment that would answer that question? Yeah, it wouldn't be too difficult, as long as you had the right
Rats.
equipment. All right, so what kind of research would we have to do before we can set this up? What are some things we would want to find out in advance before we start working on this experiment? Tyler?
What type of food would it feed them?
Yeah, we need to find what type of food to feed the rats. What else do we have to know before we can? Tyler?
The weights of the rats prior to the experiment, and like also the average weight gain, especially for the young. I don't know how much rats grow, but I assume it's a good bit.
Okay. Well, I might not know that ahead of time before the experiment. What else do you have to know?
Their habitat.
So, like, how do you keep rats? How, what kind of cage do they need? What kind of stuff goes in? What temperature should you have the room at? There's some general information that's going to help you to set up your. What are some sources of vitamin C? Where can I get vitamin C? What form does it come in? Those are things you'd have to know about before you can actually set up this experiment. So the question we're asking is, do mice gain weight if given extra vitamin C? Who could give me a possible hypothesis? What would your hypothesis be if you were setting up this experiment? I see. So give people a chance to think about it for a second before I call on it. Diksha?
Don't say no because vitamin C doesn't contain fats or not.
Okay. So give me in like a sentence. So what would you, so your hypothesis, your prediction, so you predict,
That the mice will not gain weight because vitamin C doesn't contain fats.
okay, good. Yeah, and you don't even really need the because part. So to have your hypothesis, you could just say, if mice are given extra vitamin C, they will not grow large. That would be a fine hypothesis. So again, you're predicting what's going to happen in the experiment. I have a different hypothesis here, but it doesn't matter. I said if mice are given extra vitamin C, then they will weigh more. But you could have the opposite. Like Dixon just said, maybe your hypothesis is they wouldn't. Your hypothesis always has to be a statement. It can never be in the form of a question. Because it's a prediction. It's what you think is going to happen. So how would I do this? It's time for my experiment. We have our problem, research, hypothesis. Now I need to set it up. Anyone have ideas about what would I actually do? Jack?
Well, you would feed them like regularly too, but just implement a little bit more vitamin C.
Okay. How many mice would I have?
One.
Okay, I have one mouse. I would give it regular food and extra vitamin C. Okay. Let's say we did that. And then I measured the mouse's weight after 2 weeks. Would I know if giving them vitamin C made them grow larger?
No.
What am I missing?
Control.
Yes, I'm missing something. So let's improve upon this. Ella, what does that mean, a control group? You're correct. We can need a control group. But what does that mean, though? Ashley? Okay, so how would we set up a control group? What would we do? Yeah, so you're right. We need a control group. Can't just have one mouse or one group of mice and give them extra vitamin C. We need a control group. Okay, so now we're getting there. So we have two groups of mice. Like Ashley said, one's getting just normal mouse food, the other's getting the normal mouse food, and some extra vitamin C. What else do I have to be sure of with my two groups of mice? Is that they need to get the same amount of food? Anything else? Colin?
Their living conditions, like their habitats and how much, I guess, how active they are. I don't know how
Okay.
mice would get exercise, but I'm sure
Yeah.
that would play a factor.
Basically, I want everything to be as close to the same as possible. I only want there to be one single thing different, and that's whether or not they're getting this extra vitamin C. So, set up my two groups. One group gets a normal amount of vitamin C that mice actually get, and then the second group gets twice as much. There's other ways you can set it up, but this will do for now. That leads us to the observation step. So what would I be observing if I was doing this experiment? Abby?
The weight gain of the mouse who was getting extra vitamin C, and then you would want to compare that to the amount that the other mouse was getting. He was getting more.
Correct. Yep. I would just be observing their weight. Okay, I would measure the weight of the mice getting regular food, and then observe the weight of the mice getting extra vitamin C. Those are my data that I would be collecting. So let's say we did that. After 2 weeks, the vitamin C eating mice weigh the same as the non-vitamin C eating mice. That brings me to our final step of our scientific method, which is the conclusion step. So what would you conclude? So your conclusion step, you determine, was your hypothesis correct or incorrect? So based on what I have, give me a conclusion, Tyler.
That my student not gaining weight if you feed them extra vitamin C.
Okay, yep, I could say my hypothesis was incorrect. Okay, that's all I really need to say. Does that mean I should go and erase it
No.
and steal Diksha's hypothesis? No, it's okay to have an incorrect hypothesis. You still have learned something valuable. You've still learned that vitamin C does not increase the weight of plants. So you don't have to change it or erase it. In fact, you shouldn't. You should keep it. So that's how we use the steps of the scientific method. Do you guys ever use the scientific method? When do you use the scientific method?
In like everyday sort of stuff, or just,
You do?
or I'm saying, like, what is it, like, when, do I use the scientific method if I'm doing something like this, or just like normal?
Just in general, in your everyday life. Ali?
Last time I used it was when I was testing my cereal. Is it better to put in the milk before or just after?
Okay. So that's an experiment?
Oh yeah, it tastes so much better before.
Yeah.
So you do use this all the time. Maybe it's in the case of your cereal. Maybe it's in the case of rats
As in
living
food,
under your bed.
I'm more than
Or if it's you playing video games. Okay? You experiment. Okay? Maybe you see if landing in a certain spot when you're playing Fortnite allows you to find more chests and get better weapons. And you experiment. You make a decision, you try it out, you collect some data, you have a hypothesis, you make some observations, and come up with a conclusion. If you're playing sports and you decide, well, I'm going to see if I strike the soccer ball in this way, how does that affect the flight of the ball? Okay, if you're playing music, maybe you think if I hold my hands in this different position, maybe I'm going to be able to play this scale more efficiently. Okay, you can experiment all the time. Maybe it's making you breakfast here. But those experiments, you might not write out all the steps and say, okay, my problem is, where am I going to land? I need better weapons, But really, you're doing the same process. Okay? Anytime you're trying, we call it trial and error sometimes. You're really experimenting. You may not define it and write out all the steps, but it's really what you're doing. All right, let's skip this Mario Galaxy. All right, we need to get through this so you can do your homework. So when we are talking about experiments, one important factor is something that we call variables. So, variables are factors in your experiment that you can adjust, that you can monitor, that you can sometimes change. And there's three categories of variables that we need to be aware of. They're called independent variable, dependent variable, and variables held constant. Have you guys ever heard of these? Oh, so you know these?
No.
You've heard of them, but you don't know exactly what they are. So does anyone know in an experiment what the independent variable is?
Is it the independent variable, like in the case of the lilies and the rose, the independent variable, the blue and the red?
Yeah, exactly. So it's the thing you're testing, the thing that you are in control of. If it's a good experiment, there's only one independent variable. To help you remember, I remember if it starts with an I, an independent variable, which looks like a 1 because it's the one thing that's different between your groups. It's the thing you're testing, the thing you're sort of interested in. So if you're making sure in your two groups there's only one thing different, what about all the other things? What do you have to do with them? All the other factors.
Sure they're the same.
Make sure they're the same. And those things we call variables held constant. So with the plants, once you guys fix the experiment for me, the independent variable is the color of the light. Variables held constant are all the other things. The type of plant, the amount of time, the water. So that leaves us with the dependent variable. In an experiment, the dependent variable is the data you collect, the observations you're making. So in the plant color experiment, what was the dependent variable? Growth of the plant. You don't know what it is ahead of time. It's what you're observing as the experiment's going along. Let's think about the rat experiment. In the rat experiment, what would be the independent variable? The independent variable. Let's see. Tyler?
It'd be if they would gain weight or not.
No? Lucia? Yes, the one thing that was different between my two groups of mice, the thing I'm testing, is whether or not they got vitamin C. What would be a variable I would have to hold constant? I'm sorry, I don't know your name. Nate? Go ahead.
The habitat and the amount of food you
The amount of food, the habitat, the water, the variety of rat, all those things. Good. What would be the dependent variable from that rat experiment? Big truck?
The weight of the mice.
Yeah, the weight of the mice. That's the data I would collect. Those are the observations I made. All right, last thing for today, groups. So you guys told me earlier we needed a control group. When you're setting up an experiment, you always have to have at least two groups, a control group and an experimental group. The control group is the group when you're sort of treating it normally. You're not changing the, oh, I'm sorry, we'll start with experimental. You're changing something about it. You're treating it in some way. Versus the control group is kind of just like a test run. It's the group you kind of keep under normal conditions. You don't change anything about it. It's treated normally. Sorry. So in my rat experiment, which is the experimental group? Experimental group. Sean? Sorry, I'm going to Sean.
The experimental group is the rats are getting more vitamin C.
Yep. What's the control group? Sean? Yeah, that's just getting normal food. Now you might have several experimental groups. You could have had a group of mice that got double the normal vitamin C and triple and 4 times. So you could have multiple experimental groups, but you always need that control group so you have something to compare the experimental group. All right. Any questions about what we covered today? All right, you are today's video. Let's see.